Alrighty, hello guys, welcome to my biochemistry presentation on the psoriasis biochemical pathways. So you're probably thinking, this seems actually pretty related to microbiology, and those of you in micro with me would probably agree. However, we've also learned a lot in microbiology that strongly correlates actually with biochemistry. So, for example, this is going to be a lot of autoimmunity kind of stuff, but we actually talk a lot about different antigens and antibodies and haptens and things like that. So, this is actually super relevant. And we're really going to get into the meat of the biochemistry of these pathways. And I also chose this because my boyfriend actually suffers from from psoriasis. And so it's pretty personal because I see him struggle with it a lot. And so I wanted to learn more about it so we could figure out ways I could better help him. So anyways, moving on with that. First, I wanted to mention that obviously this is not my research. And so here's the title of the research article, Inhibiting Protein Kinase Activity of Pyruvate Kinase M2 by SIR2 Deacetylase Attenuates Psoriasis. And here's the authors, Howe et al. Okay, moving forward. Let's talk about the background. Like, what is psoriasis? So psoriasis is really similar to things like eczema, etc. It's got some of the general characteristics of especially raised plaques. This is super prevalent in people with psoriasis. They have a very scaly surface. The skin underneath of the plaques is very red and tender because it's baby skin, it's brand new, and it's very sensitive. And actually, psoriasis can be an indicator for other severe illnesses such as diabetes and also depression and a couple other really important things that can be affected by it, or this can be an indication that you have one of these other diseases. So the causes of this, it's mostly unknown. However, it is autoimmune, so it's something that your body does to itself, recognizing self-antigens, which is not great. Um, so there is a specific T helper cell chemical pathway that they've been doing some specific research with to kind of pinpoint that as being one of the major causes of psoriasis in these individuals. And also IL-23 is a really important cytokine that I'm going to discuss, and so is STAT3. That is a really important protein that's involved in this biochemical pathway. So the main hypothesis of this article is focusing mostly on the role of SIRT2, which I'm going to call SIRT2, deacetylase, which is an important enzyme that deacetylases PKM2, which is protein kinase M2, in a specific type of T helper cell, type 17, which is cell-mediated inflammatory response, and see how that affects psoriasis as a whole. So here's an awesome diagram that I absolutely love. It's a very good summary of kind of what they're looking at. So if you look on the left, that cell is a normal T cell, and it has some receptors that cytokines IL-6 and IL-23 can bind to. IL-23 plays a pretty big role in the synthesis of STAT3, which is that yellow oval near the bottom. So what happens is SIRT2, what it does is it deacetylates the acetyl group from protein kinase M2, and it takes that off and it puts it on a lysine. So that actually inactivates PKM2. So when it goes into the nucleus, it's not really doing anything. So STAT3 is not activated. And so if you look over to a little bit to the right, you'll see IL-22 and IL-17, which are two really important cytokines that activate other T and B cells that are really important in the function of the immune system. And so those cause a major immune response for epithelial cells. So you'll see that's kind of lower as compared to if we move on over to the right T helper cell. We'll notice that the difference between this is that SIRT2 is crossed out, saying this is a cell that does not have this specific enzyme. So what happens is PKM2 is acetylated, and it stays that way, which it means it's active. And so it goes into the nucleus, and it sees STAT3, which once again is it's transcribed because of the binding of IL-23. And so STAT3 is phosphorylated by PKM2, and it goes on to translate some other genes like RORC. Those actually cause specific production of IL-22 and IL-17 cytokines, which once again, the larger production of those means a huge immune response. The more production of those, the bigger the immune response is to come. So basically, STAT3 plays a huge role in this pathway, whether it being activated or not. And so if you look at the diagram on the top, the blue triangle and the red triangle, there is a inverse proportional relationship between SIRT2 and PKM2 that is acetylated, meaning it is active. And so as SIRT2 levels go down, ACPKM2 levels go up, meaning that STAT3 phosphorylation goes up, meaning that these cytokines are being released, causing this immune response to attack self tissues. Okay, so going forward from that, we get into our methods. Like how are they measuring this? What do they do to come to these conclusions or like test this theory? So they had some animals, and these animals were mice that came from the Jackson laboratory. So these mice had their SIRT2 genes knocked out. That's what KO means. So knocked out. So they didn't have that. They also had recombinant adenoviruses that they used to reintroduce SIRT2 genes back into the mice that had those genes knocked out. They had several skin inflammation models and histology stains. So they took pieces of skin from the backs of the mice and they actually stained them to look at them histologically. There's flow cytometric analysis where they took cells and they put it in a salty buffer. And they actually ran lasers through it to do a couple different analyses that way. Cytokine detection, which is basically just doing some ELISA assays to detect those specific cytokines that they're going to be wanting to look for the levels of. And they also did cell culture transient transfection, antibodies for Western blots. We've actually discussed what Western blots are, so I want to explain that. The antibodies, doing different probes for PKM2 and the different SIRTs that they were studying, as well as some bird U assays. Those are some very specific assays that they used in this, but it's not super relevant, so I'm not going to discuss that. Co-immunoprecipitation is where they precipitate out some of those different proteins, and then we can probe them that way, similar to like a Western blot. Biochemical analysis, RNA isolation, so taking the RNA out of these and doing some real-time PCR, so basically flagging them with fluorescence so we can actually count how much DNA is being produced or RNA is being produced. And also we needed to be able to do some differentiation between some of the T cells. And so what they're looking at is specifically T helper cell, which is CD4 positive, and type 17 in this case. So moving forward, here is, I thought, the most relevant figure 6 from the article. Up in the top left corner, we have some histological samples. So we have wild type mice, and we have KO, which is knocked out. So that's the genes being knocked out. And then IL-23 and also vehicle was added to both of these. And when you look at the knocked out with IL-23, you can see some pretty specific psoriasis indication on these skin tissues, which makes sense. So looking forward, there are some other histological samples that once again kind of reaffirm that when you knock out the gene SIRT, you're going to see a higher production of IL-23 because that's the cytokine that's produced from that chemical pathway, as I discussed. And same with basically these other graphics are showing the same thing. This blot down here has, if you look at under wild type and under SIRT2, you can see that there's pretty thick blots there indicating that SIRT2 is present. And then if you look next to the knocked out, that's just like a reaffirmation that it was not present in those rodents with knocked out genes. And going forward, they were basically able to get some specific results. This is kind of a summary of that. So SIRT2 deficiency increased the amount of T helper cells in this type 17 that were actually activated, producing cytokines IL-17 and IL-22, which again was causing the skin cells to be attacked by these specific T helper cells. And we also were able to gather that SIRT2 is able to negatively regulate Th17 cell differentiation. So once again, going back to that STAT3. So SIRT2 has a kind of an indirect direct effect on the production of that, which allows these T helper cells to be differentiated out. So we also noticed that SIRT2 mediated PKM2 deacylation can be used as an effective option for psoriasis therapy. I mean, think about it. If you're giving people these SIRT2 proteins, these deacetylases, then they can regulate that PKM2 phosphorylation. So we can kind of down regulate that and decrease the amount of cells that are being attacked by these T helper cells. So the main significance with this is, treating psoriasis is that would be a huge advance because it's one of these autoimmune diseases that people don't really understand. So it's a pretty big step in science to be able to finally understand some of these autoimmune diseases. And also there's just so many people that's struggling with it. It's embarrassing. It can be tricky to deal with. Once again, it can cause other major health problems when left untreated. So being able to treat this and kind of like retrain the immune system or kind of find ways to treat the immune system when it starts attacking self antigens, it's really important for science. And I think that it also once again is a huge step because then maybe we can start looking at other ways that autoimmune diseases function. And thank you for listening to my discussion on skin disease because I absolutely love skin disease. So thank you for being a part of this with me. I'm glad I could do something that was super relevant to what I enjoy studying as well as the biochemistry of that. And here's the official citation just for kind of finishing that, wrapping that up with a bow. Anyways, thank you for my presentation, listening to my presentation. And if you have any questions, feel free to ask me in class or email me. And yeah, that's basically it. So thank you so much for listening.